Hello everyone and welcome to Lab Manager's Product Spotlight webinar series. These webinars are designed to help attendees understand a specific proprietary service or technology and where and how it can be applied in an effort to help labs operate more efficiently. My name is Trevor Henderson, Technology Editor for Lab Manager, and I'll be moderating today's webinar entitled Molar Mass, Size, Charge, and Interactions, Light Scattering Tools for Essential Biophysical Characterization. We like our webinars to be very interactive, so we encourage you to send us your questions at any point during the presentation. Many of you know the drill, but for those of you just joining us, you may simply type your questions by entering them in the query box located at the bottom left-hand side of your screen. We'll try to address as many questions as possible during the Q&A period. However, if we happen to run out of time, I will forward any unanswered questions to our presenter, and he can respond to you via email. Additional resources can be accessed through the resource widget located at the bottom of your screen. You may also move or resize any of the windows simply by grabbing them at the top or stretching them at the bottom right corner. At this time, you may need to move or minimize some of the open windows to see the live view. This webinar recording will be available early next week on Lab Manager's website. At the end of this webinar, I'll share that link with you, as well as the contact information for our speaker. At this time, I'd like to introduce our speaker who joins us today from Wyatt Technology. Daniel Sohm is Principal Scientist as well as Director of Marketing at Wyatt Technology. He's been with Wyatt for over 10 years, first in the R&D and then in the Marketing Department. Prior to joining the scientific instrumentation world, his professional endeavors included the semiconductor and defense industries. Dr. Sohm completed his undergraduate degree in physics at the Technion Israel Institute of Technology, his doctoral research in the Brown University Physics Department, and postdoctoral research at Los Alamos National Lab and the Weizmann Institute of Science. Thank you for joining us today, Dan.
Well, thank you very much for the introduction, Trevor, and thanks to all the attendees for joining in. I hope I'm able to present something of interest to you. And so let me jump right in. As Trevor mentioned, the topic of our talk today is molar mass, size, charge, and interactions. You may not normally associate some of these essential properties of macromolecules and nanoparticles with light scattering, but as we'll learn today, there are some really useful tools associated with light scattering that can help you assess these properties. And so if you're carrying out any type of biomolecular research, you go through a cycle similar to the diagram that I have on this slide. You're going to make something, proteins, there might be some other types of macromolecules or nanoparticles, and you need to ask yourself certain questions along the way. Did you make what you intended? What are its essential properties, conformation, molecular weight, size? How is it aggregated? Perhaps it's conjugated. Perhaps it's a glycoprotein or otherwise some type of conjugate of, say, a protein or polysaccharide. Does it do what it's supposed to do? Does it have the proper interactions with a target or with an effector? Is it a well-behaved molecule? So how does it self-associate? Does it become viscous when you use it at high concentrations? How does it interact with any excipients in the formulations? And finally, is it a robust product? Is it stable? Can you make it regularly lot to lot and come up with the same properties? And so the acronyms that are in this diagram are all light scattering based techniques, and we'll go through each one of them to learn a little bit more about them, so I won't belabor now what they all stand for. But the idea is that each of these techniques, as we go through them, can be applied to answering these critical questions. So the first type of light scattering is multi-angle static light scattering. Multi-angle light scattering, or MALS as I like to call it, is a first principles technique, and it simply relies on shining a light, usually a laser beam, through a solution and measuring the scattered intensity, often measuring it as a function of angle, which is where the multi-angle part comes from. We measure it over several angles. And the first principles relation that arises from thermodynamics, essentially, is that the intensity of the scattered light is proportional to the molecular weight of the molecules in solution times their concentration, where the concentration is the weight concentration, grams per mole. It's very simple and very powerful. because if we measure the scattered intensity and we measure the concentration, we can determine the weighted average molecular weight of the molecules in solution. It's also possible to determine a measure of size called the root mean square radius. Without going into that deeply, that essentially gives you the average position of the mass in the particle relative to the center of gravity of that particle. It's a measure of size, which is, again, highly useful, and determined from first principles. Through these first principle analyses, we can address some derived measurements, including interaction parameters. So what is the stoichiometry of the complex it forms? What is its KD, its equilibrium dissociation constant? Rates of association, rates of aggregation, rates of dissociation, and also another measure of interaction called second viral coefficient, typically known as A2 or B22, which is a measure of nonspecific interactions between molecules. Because MALS is first principles, it's really important as an in-depth characterization tool. Now there's another type of light scattering, another flavor as I like to call it, which is dynamic light scattering, also known as quasi-elastic light scattering. And in DLS, instead of simply measuring the scattered intensity as a function of angle, we're going to measure it as a function of time. And it turns out that there are fluctuations over a rapid time scale of the light scattering intensity, and those fluctuations are due to Brownian motion of the particles. Basically, as particles or macromolecules are in solution, they're jittering around rapidly due to Brownian motion, and that causes these intensity fluctuations. The rate of intensity fluctuation is closely related to the rate of diffusion due to Brownian motion, and therefore dynamic light scattering from first principles determines the diffusion coefficient, the translational diffusion coefficient of the molecules or the nanoparticles. And through the Stokes-Einstein relationship, which I've cited here, that can be converted to a different measure of size known as the hydrodynamic radius. The hydrodynamic radius you can think of as being the envelope radius of that particle. So it's a slightly different measure of size than the RMS radius, or the radius of generation. One of the nice things about dynamic light scattering is that you can get an estimate of size distributions even without any preliminary fractionation. As I'll show you, MALS is typically used with some type of fractionation technique in order to get a highly accurate size dependence and distribution of sizes in molar masses. With dynamic light scattering, you can get a coarse size distribution even without any fractionation in very little time. So it's really effective in that manner. You can get some measures of interactions. So the concentration coefficient of the diffusion parameter, diffusion coefficient, is a parameter known as KD. It's not the same KD as the as the equilibrium dissociation constant. Kind of unfortunate that they were given the same name, but there's a lowercase and uppercase. It is related actually to the second viral coefficient, that thermodynamic parameter related to nonspecific interactions. So we can use DLS to assess interactions as well. And one of the really nice things about dynamic light scattering is that it's amenable to high throughput screening. So it's a little more forgiving in terms of things like stray light and other types of things that you have to be really careful about in MALS. And so dynamic light scattering can actually be done in situ in microwell plates, and so that permits high throughput screening, and that ends up being extremely powerful in assessing formulations and developability, aggregation states as a function of formulation, and so forth. And then there's a third flavor of light scattering, and that is phase analysis light scattering, or electrophoretic light scattering, which measures the speed of motion of particles under an applied electric field. And from that, we determine the electrophoretic mobility mu. And if we also measure the hydrodynamic radius, we can combine that information to determine something known as the zeta potential, or the net charge on the molecule. Now, the instrument I'll mention later on uses a sophisticated variant of the standard PALS technique, where we have 31 measurements instead of a single measurement, which is more common. It's turns out to be extremely sensitive, gives you a very high dynamic range, and allows you to make rapid measurements at reduced voltages in order to preserve your biological samples. So the tools that I mentioned earlier are essentially combinations of these different flavors of light scattering with certain types of sample preparation and delivery. The first tool I talk about combines standard size exclusion chromatography as a sample prep and delivery method to the light scattering detectors. So SEC-MALS, or I'll call it SEC-MALS for short, utilizes a MALS detector. It will often use either a refractive index or UV detector for concentration measurements, and in some cases we actually utilize both in tandem. And you can also embed into the same MALS detector a dynamic light scattering module. So we can measure a host of properties after separation on the size exclusion, and that allows us to get accurate distributions of molecular weights, sizes, useful in characterizing the smaller aggregates or oligomers that will separate nicely on standard size exclusion columns. Depending on the specific detectors that you have, can cover extremely wide range of molecular weights and sizes. Pretty much anything that you can separate on a size exclusion column can be well characterized using light scattering. Range of RGs typically goes from 10 nanometer up to, depending on the instrument, 50 nanometers, or the limit of separation of the column. RHs, the hydrodynamic radii, from 1 nanometer up to hundreds of nanometers, and the molecular weight range can also be extremely large. So why would you go to all the bother of adding these light scattering detectors to size exclusion chromatography when it's commonly accepted that size exclusion chromatography is a good way to measure molecular weights? Well, it turns out that size exclusion chromatography, when you're using molecular standards as references to establish the connection between elution time and molecular weight, it is not actually a very accurate technique. Actually, size exclusion does not separate by molecular weight. It separates by hydrodynamic volume. And so in order for those references to give you the proper connection between volume and molecular weight, they need to be very similar to the molecules that you're measuring, your own samples. They need to have the same conformation and the same density as the standards. They need to have the same types of column interactions, so hydrophobic interactions, other type of non-ideal interactions with the column need to be the same as the standards in order for them to represent correctly the properties of your molecules. In addition, your chromatography needs to be reproducible. So if there are some variations in, say, aging of your column over time, you need to recalibrate on a regular basis. Actually, light scattering obviates the need for all of those assumptions and gives you the information on molecular weights directly and independently of elution time. In the graph that you see here, we see separation of BSA and hemoglobin. Now, the BSA, of course, is one of those references that you typically use for column calibration, and it does not have any non-ideal interactions with the column. Separates nicely. You can see the monomer and dimer peaks here, and the dots that are in those graphs are the molecular weights as determined by light scattering. And so light scattering gives you the correct molecular weights. If you then look at the hemoglobin peak, you see that it elutes much later than the BSA monomer. And nominally, you would expect that that means that the hemoglobin has a much lower molar mass than the BSA monomer, but of course that's not true. Hemoglobin has a molar mass which is approximately the same as the BSA monomer. The light scattering, however, which is the blue dots along the peak, gives you the necessary information. It gives you the molecular weight across the peak, and it gives you, and you can see that on the larger time where there's this kind of funny shaped shoulder on the hemoglobin peak, the molecular weight decreases based on the light scattering information. The reason that it gives you this decrease is actually because hemoglobin is in dynamic equilibrium between monomers and tetramers, and as the concentration decreases, you get fewer tetramers and more dimers, and so the overall molecular weight decreases. So light scattering gives you all that information. It shows you how the molecular weight actually varies across the peak, regardless of elution time. This is a nice example of insulin in some different formulations, and you can see the information that we obtained from the light scattering the molar masses. In the red, we see an insulin formulation which is primarily monomeric, and the light scattering indicates that the molar mass is flat across that entire peak of the monomer, so it's perfectly monomeric. And then there's a small shoulder at the earlier elution volumes, and the light scattering again shows us that goes up to the molecular weight of a hexamer. The blue formulation, sample number 2, has primarily hexamers. That's the peak at about 14 milliliters. And it has an additional peak later on at about 16, 17. And the light scattering shows us that the molecular weight changes over that peak. Again, that's due to dynamic equilibrium between monomers and dimers. And we can see that all clearly, even at these low molecular weights. Light scattering works very nicely at high molecular weights as well. And so here are some relatively large protein molecules, apoferritin and IgM. It's interesting to note that the dimer of the apoferritin and the monomer of the IgM have approximately the same molecular weights, but they elute at very different times. And so you can see the monomeric peak of IgM in the light scattering calculation is about the same as the apoferritin, but very different elution times. Light scattering doesn't mind that because it doesn't care about elution times, and so it becomes very powerful in these types of analyses. There we go. If you look at the left side of this graph, you see in the red trace of the light scattering a small peak, of which there is no evidence at all in the concentration trace, which is the blue. And what's happening there is that there are some relatively large aggregates eluting. Because light scattering is proportional to the product of molecular weight and concentration, it's going to be much more sensitive to larger aggregates than a simple concentration signal, and will indicate those even when you don't see them in the concentration signal. So far we've talked about a standard type of light scattering molecular weight analysis. We have 2 detectors, light scattering and concentration, and essentially the ratio of light scattering to concentration gives you molecular weight. We can extend that to a multi-detector technique. We're using information from 3 detectors, the light scattering detector, a UV detector, and the refractive index detector, to obtain additional information for conjugated molecules. The idea here, to break it down to its simplest form, is imagine that you have, say, a glycoprotein or a PEGylated protein, where you have protein which is sensitive to UV. The UV detector will give you the concentration of the protein. The refractive index detector is actually a universal detector, so it gives you the total concentration of all the macromolecules, say the polysaccharide or the PEG, as well as the protein. And so between the 2 of them, you can figure out how much protein you have and how much of the conjugate you have. Add to that information the light scattering, and you can determine the molecular weight of each of those components in the molecule. And so what we see here in the graph is the red trace, which is the total mass of the conjugate, the green trace, which is the molecular weight of just the protein. So we can determine, for instance, if the protein is monomeric or oligomeric. And then the total molar mass of the modifier, may that be the total amount of glycans or the PEG or some other modified molecule. So here's an example of how we can use that to assess the difference in glycosylation state of a recombinant protein expressed in different host cells. The mammalian cell, which is the blue, and the insect cell, which is the red. And the analysis shows us that the protein molecular weight in this glycoprotein is identical between the 2 of them, which is what we expect because it's the same protein being expressed in different cells. But the degree of glycosylation is different between the 2 cells. There's more glycosylation from the mammalian cell than from the insect cell. And if you look at how the molecular weight of the conjugate of the carbohydrate changes across the peak, you can also get an indication of the polydispersity of the glycosylation in the different cells. The same type of analysis can be used to analyze antibody-drug conjugates, and here the molar mass is on a logarithmic scale. So you see that these two ADCs, which have same antibody but different drug and linkers, have identical molar masses of the antibody, different molar masses of the conjugate, and if we know the molar mass of the drug linker combination, we can simply divide the total molar mass of the conjugated drug by the individual molecular weight to get the drug antibody ratio, the DAR, shown in the lower right of the table. So in this example, if we were looking at a protein-nucleosome complex and looking just at the chromatographic trace, we see 3 peaks, and we might nominally expect that we have a monomer and some aggregates. The light scattering tells us, though, that the molecular weights of all 3 peaks is the same. And so what's going on here? Why are we getting different elution times from these 3 different species, which all have the same molecular weights? Well, in this system, we've added in the dynamic light scattering module, which gives us the sizes of these different complexes. And what we find is that the different peaks correspond to different hydrodynamic sizes. And that indicates that what probably is going on is that we have some partially unfolded protein-nucleosome complexes. And those which are unfolded have a larger hydrodynamic volume and are eluting earlier on. Some other things that you could infer from this or not infer, depending on the results, are the different elution times related to different hydrophobic interactions or non-ideal column interactions, or it is in fact just due to different sizes. And finally, we can use the SEC-MALS to assess protein purification. And so here we see a monomer and some aggregates. And if you look at the right-hand shoulder of the primary peak, there's a little bit something going on, possibly tailing due to non-ideal interactions with the column, or possibly some contaminants. Applying the light scattering analysis, we find out the molecular weights of the aggregates, and we also find out that that little shoulder actually has a molecular weight of 42 kilodaltons, and so it is perhaps some fragments or perhaps some host cell proteins, but not just simply the monomer sticking to the column. So this helps us understand what we're getting off the purification. Sometimes you have tightly bound oligomers, and so as they go through the dilution and shear of the column, regardless of the concentration that you inject, you'll get the same molecular weights, and that's indicated here. So this was a stable oligomer, but sometimes the oligomers are in a relatively loose dynamic equilibrium with the monomers. And so as you change the concentration, you'll get a different equilibrium ratio between monomer and oligomer. And the overall molecular weight that we measure with light scattering will change. So it will change with concentration. We see that across the peak here and also between different injections which have different concentrations. And so this is an indication that there's some reversible interaction occurring in the molecule. It's a good way to get an initial estimate of what's going on. If you actually want to study the self-association that's indicated here, then we have another technique which I'll describe later on called CG-MALS. But this is a good way just to get an initial indication through the SEC-MALS technique that there is this reversible interaction going on. The detectors for MALS have until recently only been applicable to standard HPLC, standard SEC, GPC, but recently a new detector called the microdon was released, and this microdon is actually useful with UHPLC in order to confer all of the benefits of the SEC-MALS analysis to the advantages that we get with UHPLC, the higher resolution, reduced mobile phase consumption, and faster throughput. So to summarize, SEC-MALS and also the SEC-DLS technique, which is the online DLS, give you Absolute molar masses and sizes in solution without reference to molecular standards and elution time. They overcome the limitations of the standard SEC. Will give you positive identification of aggregated and unfolded species as well as impurities, and a particularly powerful analysis that can be carried out with SEC-MALS. The conjugate analysis gives you the PEGylation state, glycosylation state, or DAR. Now the next tool that I'll describe is FFF-MALS. FFF-MALS is conceptually very similar to SEC-MALS. It's a separation technique which is combined with light scattering downstream of the separation. But sometimes SEC is not a suitable separation technique depending on your particular molecules or nanoparticles. It might be because of the size range that you're looking at. It might be because of column interactions. And often you can overcome those separation challenges using a field flow fractionation technique. And it's applicable both to macromolecules and colloidal systems. The Eclipse system that Wyatt offers integrates with the standard HPLCs that are used in most labs, Agilent, Thermo, Shimadzu, and others. And the salient property of field flow fractionation is that it will separate an extremely large size range from 1 to 1,000 nanometers. Downstream of that, we can put the light scattering detectors to get the additional characterization, regardless of exactly when things elute. Otherwise, the properties are measured are very similar: molecular weight, size, conjugation, aggregation. So briefly, how FFF separation works: instead of having a packed column, we have a very simple channel, which is just 2 plates with fluid in between. The top plate is just a solid plate, and the bottom interface is a frit, which supports a semi-permeable membrane, very similar to a dialysis membrane. The solvent is just made to flow parallel to the channel, but we restrict the flow at the output, and that forces some of the solvent to flow through the semi-permeable membrane, and that is called the cross-flow. The cross-flow will push the sample against the membrane. But then there's another force acting on the sample, which is Brownian motion and diffusion. So depending on the size of the sample, it will back diffuse up the channel to different heights. The large particles will not diffuse very much, and they'll stay close to the membrane. The smaller particles will be able to diffuse higher up. Now the channel flow has this parabolic flow profile, and so the higher it diffuses, the faster it's going to elute out the end of the channel, and this is how we affect the separation. The smaller things go higher up into the channel, are swept out first. The larger things stay closer to the membrane and are swept out later on. So it's a very gentle technique. There's very little shear involved, very little surface interaction, and has many advantages over standard packed column separations. This technique will give you separation even for macromolecules, which is as good as or better than size exclusion. Very large range for separations. And one of the really cool things about FFF is that you can optimize the separation power for different ranges simply by adjusting the flow parameters, that is the ratio of the cross flow to the channel flow, and a geometric parameter, which is a spacer that you put between the 2 plates. In fact, you can even adjust these things on the fly. You can adjust the flow rates, at least on the fly, to change separation conditions as you go. Open channel, little shear, works under most native conditions, and will eliminate adverse interactions with the stationary phase. FFF is recognized by the FDA as an orthogonal method to SEC for characterization of aggregates, and so if you are working in a lab which is responsible for characterizing biotherapeutics, it's a useful technique to have in your arsenal. So I mentioned that you can tune the flow parameters in order to achieve different separations. In this example, what we did was we zoomed in through those parameters to achieve very good separation of oligomers here for an antibody. You can also change the parameters to zoom out and look at a much larger separation range. So here we're looking at a much larger range of oligomers, aggregates that are going up into the tens of millions, and still get a nice separation across that entire range. And in fact, you can even change things as you go so that you're zooming in on different ranges at different parts of the separation. This is an example of a colloidal system. So it's a antimicrobial system using silver as the active agent, and the microsilver is being carried by these nanolipids to which it's absorbing. And the question being asked here was, using different preparative conditions, different ratios of nanolipid to microsilver in the preparation, how does the size of these particular colloids change? And so what was done was do a separation on field flow fractionation, use moles downstream as the sizing technique, and you can see that depending on whether you have pure nanolipids or different amounts of microsilver bound, you can see very clearly very small changes in the size, on the order of a couple of % in the size due to the different formulation conditions. I don't show it here, but this is actually a very robust technique, so if you run the sample multiple times, the overlays are far better than the distinction between the different conditions. So the small changes that you see here due to the different preparative conditions are in fact real and extremely accurate. So if you're working on these colloidal systems and you need accurate size analyses, the combination of FFF with MALS is an extremely powerful technique to get accurate and sensitive measurements. This is an example of a virus-like particle. In the course of its processing and preparation, there was in solution a mixture of well-formed virus-like particles and some loose products or subproducts, and these were nicely separated on FFF. You could obtain both the molar mass and the size. And this is an example of separation of two virus-like particles which are very close in size. One is 16 nanometers, the other 18.5 nanometers. FFF gives a great separation, and the light scattering signal on top of that, the analysis that we get from light scattering, gives molecular weights and sizes. And then finally, conjugate analysis can be applied to FFF separations as well. And in this example, we're looking at a virus-like particle, which is bearing a DNA payload. And again, through the 3 detector technique of the UV plus RI plus MALS, we can determine the overall molecular weight of the VLP bearing its payload, as well as just the protein part. The FFF technique that Wyatt offers, the Eclipse system, offers a wide range of separation channels, which allow you to do separations ranging from nanograms up through the milligram quantities on the semi-preparative channel. So it's extremely versatile, and there are a wide range of options available for achieving different analytical separations. To summarize, FFF-MALS is going to be your primary tool for molar mass and size analysis of proteins and other macromolecules that don't elute well on SEC, as well as for nanoparticles and submicron aggregates. In particular, if you have complex fluids, which combine both macromolecules and nanoparticles, it's probably the only way to get a proper separation and sizing. And in addition, you can even couple the FFF to ICP-MS if you need elemental analysis. The third tool I'd like to address takes the same MALS detectors that we've looked at so far in conjunction with SEC and FFF and utilizes a different sample preparation technique. Here, instead of looking at distributions of molecular weight and sizes, we're going to be characterizing interactions. So the composition gradient multi-angle light scattering system, or CG-MALS, that is realized in the Calypso system is a powerful technique and versatile technique for looking at interactions without labeling and immobilization. Because it is a first principles technique and can characterize both simple and very complex interactions, a wide range of KDs from sub-nanomolars to millimolars. Typically we use it to look at equilibrium, but it is a stop flow technique and can be used to assess kinetics, but not very fast kinetics, kinetics on the orders of seconds and longer. And typically can be used to characterize both specific interactions and the non-specific intermolecular interactions that are important in understanding stability in formulations. The properties that are measured in CG-MALS include affinity, stoichiometry, and by stoichiometry, I don't mean just the stoichiometric ratio, which is how many moles of one molecule bind with how many moles of the other, but the absolute molecular stoichiometry, how many molecules of one protein bind with how many molecules of the other protein, how many are included in the oligomer, and so forth. So CG-MALS gives you the absolute stoichiometry of your complexes and reaction rates, as well as second viral coefficients. So how do we measure self-association? If you think of a self-associating system, when it's present at low concentration, it's mostly going to be monomeric, and as you increase the concentration, you're going to have formation of more and more oligomers. And so what we do is we use the CG-MALS system to prepare some different concentrations and inject them into the MALS detector. It's a stop flow technique, so they're basically prepared by the Calypso and injected into the MALS detector. The flow stops, and we make the measurements. Here's what you might expect for a simple self-association. So if there's no interaction, if there's no association, that's the blue line, the simple linear relationship between the light scattering signal and concentration. But if there is an association, as the concentration increases, the molecular weight will increase, and you'll have a nonlinear relationship between light scattering and concentration. The green line here is what you might get, for example, for a 10 micromolar dimer, and the black line is what you might expect for a 1 micromolar dimer. And the black line has a higher light scattering than the green line, just because more dimers will form at a lower concentration, and so the average molecular weight that you measure in light scattering is higher. The red line here is a 10 micromolar trimer, and you'll notice that it has a higher signal than the 10 micromolar dimer, again because the trimer has a higher molecular weight. So as those trimers form, the light scattering will increase more rapidly than for the dimer. The red line also has a different shape than the green line, and that's because the concentration dependence of the formation of trimers is different than that of dimers. And so by analyzing those relationships, we can extract the KD and the stoichiometry of the interaction. When you go to much higher concentrations, actually some funny things happen, and I won't be delving into that in this right now, but if you're looking at concentrations of tens and hundreds of megs per mil, very interesting things happen in light scattering, and you can extract a lot of interesting information. To look at heteroassociations, normally, when you do heteroassociation analysis, you think of titrations where you have a fixed concentration of one protein, and you titrate in the second protein, and you get the sigmoidal curve. That's nice, but it turns out that in composition gradient MALS, we can use a more powerful gradient, and that's what I call a crossover gradient, where we probe a larger range of stoichiometric ratios in order to get more information and more intuitive and useful information about the interaction. And so, again, if there's no interaction, the light scattering signal will just be this linear relationship across the gradient. But if there is complex formation, then somewhere around the middle of this, where you have the right stoichiometric ratio, you'll get the most complexes forming, and therefore the highest molecular weight. And that will give you the peak in the light scattering. The tighter the complex, the more complexes form at a given concentration, the more light scattering, and the higher the peak, and that's how we can analyze the KDs. Now, these graphs are simulations just to help us understand what's going on. The simulations are actually provided in the software to help you design your experiments. If you were asking, well, how do I see different stoichiometries? The position of the peak along the gradient will tell you about the stoichiometry. So 1 to 1, 2 to 1, 3 to 1, the ideal position for formation of complexes will shift along that gradient. You'll notice that the light scattering signal also increases, and that's because the molar mass of 2 to 1 is higher than 1 to 1, and 3 to 1 is even higher. And in fact, if you have a 2 to 2 complex forming, it will peak at the same position as 1 to 1, but the light scattering signal will be double that of the 1 to 1 because the molecular weight is double. And so again, this points out how CG-MALS, because it's measuring directly molecular weights, can give you the true molecular stoichiometry. This is information that most techniques will not give you. CG-MALS is carried out with this multi-syringe pump system. There's computer-controlled syringe pumps that inject different amounts of sample through filters and mixers and all the kind of preparation that you need. That's what the Calypso does, injects into the MALS detector, a DON or a mini-DON MALS detector and concentration detector. And again, the relationships between light scattering and concentration give you the molecular weights, which are analyzed in the Calypso software to analyze the KDs and stoichiometries. And so I won't go into this slide, but essentially the software offers you a very large variety of interaction models that can be analyzed. And again, because it is first principles, because we're measuring molecular weights directly, it makes it a very robust analysis. Actually, the equations that describe the light scattering signals and the concentration signals and so forth are highly constrained. And so you can't just fit any interaction to any light scattering signal. And we'll see that in the next in some of the examples coming up. In this system, we're looking at an enzyme, and it was known that this enzyme needs to be dimeric in order to be active. It was also known that the activity decreases with increasing the ionic strength. And so the question asked was, is the ionic strength impacting dimerization? Is that the reason for decreased activity? And I will go through this kind of quickly in the interest of time. But the CG-MALS analysis shows that over the different salt concentrations, you can find a region of monomer-dimer equilibrium. You can find a region of dimer-tetramer equilibrium, where all of the monomers have been dimerized, and then there's a little bit of additional self-association of the dimers. And then in this intermediate ionic strength, actually both of those are occurring simultaneously, and you can pick out from the CG-MALS analysis both the monomer-dimer association constant and the dimer-tetramer constant. Insulin tends to self-associate. If you have zinc, then it tends to form hexamers, but if there's no zinc, it self-associates differently. Here was an analysis done on self-association of insulin in the absence of zinc, and you can see how the apparent molecular weight increases with insulin concentration. If you try to fit these data to a model of hexameric self-association, that's the dashed purple line, you can see that these data just do not fit that self-association scheme. In fact, the simplest association model that fits these data is called isodesmic self-association, and that means that it forms monomer, dimer, trimer, tetramer, pentamer, hexamer, and so forth, all with a single binding constant of 52 micromolars for each new monomer that joins that chain or clump of insulin. The next example is antibody-antigen binding. And so in the CG-MALS analysis, we actually include three parts. On the left and on the right of the method shown here, we see concentration series in the individual molecules in order to any self-association. And the central region holds the heteroassociation gradient, where we're mixing together the thrombin and, in this case, thrombin and its antibody to look at self-association, heteroassociation. And so what we find after we run the measurements is that neither the IgG nor the thrombin are self-associating appreciably. We determine their molecular weights. And then in the central part, if we look at the blue line here, which is the light scattering signals, we can see clearly the injections. We can see the effect of the stop flow, where after the injection we stop the flow, and the reaction of the association between the thrombin and antibody reaches completion and reaches equilibrium. And we can also see the impact of the complex formation, which gives us excess light scattering over the dashed purple line, which is what you would expect if there's no interaction between the molecules. Now, if we naively try to fit these equilibrium data to a one-to-one interaction model, this is what we find. The blue squares here are the equilibrium data that we just looked at, and the black line is the attempt to fit these data to a one-to-one model, and you can see that the fit is very poor, and so we can rule out a one-to-one interaction. And if we then fit to the correct model, which is a bivalent model, assuming equivalent and independent binding sites, we find that there's a very good fit, and we get a KD of 9 nanomolars, which is very similar to the value that provided by ELISA, 50 nanomolars. And we can even determine from this calculation at any given composition what is the amount of one-to-one complex in equilibrium with two-to-one complex in equilibrium with monomers in that solution. I'm going to have to switch. Okay. In the interest of time, I'm not going to talk about some of the more interesting CG-MALS experiments, but you'll be able to download the slides and certainly ask me about it later on. So I'll just go briefly through this, looking at the kinetics of irreversible binding as well, and determining if there's competition between epitopes between the, in this case it was thrombin, antithrombin. Just go briefly back. Competition between of epitopes of antithrombin and an antibody for binding on the thrombin. So this is what was analyzed here. And then metacomplexes, which are multivalent, so both binding partners are multivalent. Give some really nice things. And I'm going to skip ahead. There we go. So to summarize, CG-MALS provides reliable assessment of biomolecular self-association. You get affinity and oligomeric state in solution without labeling or immobilization. Very rich information on complex interactions. And while I haven't discussed it here, it's one of the few techniques capable of quantitatively characterizing protein-protein interactions at very high concentrations. The final tool that I'll talk about, oh, I'm sorry, it's not the final tool, but second to last, is batch dynamic light scattering. Batch dynamic light scattering, either in a standard cuvette-based format or in high-throughput format using standard microwell plates, is useful for assessing size distributions and aggregation. Can also be applied to characterize colloidal stability, unfolding or aggregation onset temperature, and even viscosity of high concentrated protein solutions. Dynamic light scattering, as I mentioned earlier, works by measuring the light intensity fluctuations. The rate of fluctuation is related to the diffusion translational diffusion coefficient, and from the diffusion coefficient we can estimate an effective radius known as RH. There are analyses that will allow us to assess roughly the polydispersity. We can get the average radius of the macromolecules or nanoparticles in solution, and we can do high throughput screening, which is really useful. Okay. So, as I mentioned earlier, you can get a rough analysis of size distributions even without that preparative separation like SEC or FFF. In order to see distinct species, they have to be well resolved, so they actually need a difference in radius of a factor of 3 to 5 times. So that's something like a factor of 100 in molecular weight. Obviously much coarser than you get from a size exclusion separation. But you can see shifts in average size on the order of 1% or 2. And so the impact of aggregation, even if it's the addition of small oligomers, while you can't resolve them, you can see shifts in RH that are caused by those, as well as the changes in the polydispersity measurement, which is the width of the peak. Here's an example of looking at the impact of some different formulations and measuring the aggregation state before and after lyophilization. We can see both the very large aggregates that are formed up in the range of 100 nanometers, but you can also see these really small shifts in the primary peak, both in the width and in the average position. And those will give us a useful indication of aggregation, even if we don't know exactly how many dimers or how many trimers are formed. It's useful for a rough screening of what's going on. And so the same thing can be applied in a microwell plate. You can basically fill up a 96 or 384 or 1536 well plate with different formulations or with different candidates. If you have multiple candidates, we'd like to assess their developability. And the software allows you to color code the results to give you a nice visual indication of what's going on. And what we've done here is we've color coded the red for the nicely monodispersed samples, which do not show a lot of aggregation. The blue show intermediate aggregation, and the black show heavily aggregated samples. And you can use this both in a formulation scenario, you can use it in developability scenario. Another place to use it is if you, just as an indicator of protein solution quality. For instance, if you're doing surface plasmon resonance, doing high throughput screening of binding, you can just take your microwell plate, put it on the dynamic light scattering plate reader, which is not perturbative, it's not removing samples from the wells, it's just shining light into the wells and measuring the scattering. Assess the quality and then decide which samples you want to run in your binding study. can also ramp the temperature in both of the instruments, both the cuvette and plate-based instruments, and that's useful in understanding the conformational stability, which is indicated by the temperature onset of unfolding or aggregation. You can even use dynamic light scattering to assess viscosity of highly concentrated protein solutions, and we do that by spiking into the solutions beads of a known size. Basically ignore the light scattering from the protein and only look at the light scattering from the beads. And the diffusion coefficient of the beads will depend on their known size and on the viscosity, which is eta in this equation. By inverting the equation here, we can determine viscosity based on the diffusion coefficient of the bead. The last tool I'll be talking about is the Mobius, which is electrophoretic mobility detector. It measures electrophoretic mobility. It also measures through dynamic light scattering size. The combination of those will give you charge and zeta potential. And you can use it to assess PI in native solution, as well as the impact of molecular charge on colloidal stability, or zeta potential on colloidal stability. The Mobius instrument is compatible with auto-samplers, so you can automate your measurements. You don't have to manually change the solutions one by one. It can work at low applied voltages in order to not impact fragile molecules. And it has the additional accessory of the Atlas, which pressurizes the flow cell and allows you to work in high salt buffers, alleviating the potential electrolysis effects, which generate bubbles and interfere with light scattering. So some applications of charge measurement. In this application, we looked at heparin, and this came out of some work at the time when there was the scare of the heparin substitutes, which were coming out in some low-quality source materials. Chondroitin sulfate was this, and chondroitin sulfate is supersulfated relative to heparin, and it was thought that perhaps we could identify it through its additional charge, molecular charge. And so by examining different types of standard heparin, reference heparins, you could find out the molecular charge and its relationship to molecular weight. So turns out that all of the good quality heparins fall on this single line describing the effective charge versus molecular weight, whereas the supersulfated heparin has a more negative charge and falls off of this line. Another example is the PAMAM Dendrimer, and it's measured in very high ionic strength conditions, hundreds of millimolars of salt, and despite the high charge, the molecular charge, as it changes with dendrimer generation, comes out very clearly. So additional applications of MP-PALS, including charge states of antibody-drug conjugates, true PI determination of monoclonal antibodies, chemical stability screening, so if there's some deamidation or oxidation which is changing the charge state, that can be assessed, as well as formulation of liposomes and other types of drug delivery nanoparticles. So so I'd like to wrap this up now. Coming back to the diagram I showed you at the beginning, now we can tie it all together. We have an understanding of what all these acronyms mean. These are all the different light scattering tools, combining different types of sample prep and delivery with one of the three flavors of light scattering in order to answer your essential biophysical questions. Did you make what you intended? Does it do what it's supposed to do? Does it behave well? And is it a robust product? If you're interested in learning more, I'd like to reiterate that among the resources that you can download, there is a list of links to various pages on Wyatt's website, which has informational material about these different techniques, about the ways of measuring the different biophysical properties with light scattering, and you can explore that further, as well as view some other webinars that we have on demand, or read some white papers or application notes. And with that, I'd like to thank you all for your attention and turn it back to Trevor, and we can answer any of the questions that have come up.
Great. Well, thank you very much, Dan. I think we will go directly into our question and answer session. If you have any further questions for Dan, and you may have joined us late, you can ask your question simply by typing your query into the question box, which is located at the bottom left-hand side of your screen. First question we have, Dan, is do you require specialized plates for the DLS plate reader?
No, these are standard UV plates that you would use in a UV spectroscopic plate reader. They have transparent bottoms, but they're pretty much off the shelf.
Great, thank you. Now, how much material do you require for CG-MALS, and how does it compare to ITC or SPR?
So any type of interaction measurement, you're going to work approximately at the concentrate around KD. And so then the concentrations and total amount of material that we're using depends on the KD that you're measuring. So what I'd like to do is estimate the amount that you need for a given KD. If you're looking at typical antibody-antigen interaction with KDs of sub-nanomolar and up to tens or about 100 nanomolar, you're going to be using 100 to 200 micrograms of each protein. If the interactions are weaker and you need to measure higher concentrations, then you're going to be using more material. So if you're in the micromolar range, you might be using a couple of milligrams of material. It turns out that it's comparable or slightly less than standard ITC and a little bit more than standard SPR.
Great, thank you. Now, Dan, does MALS give the true molecular weight at ultra-high protein concentration, or would multiple scattering and other complexities complicate analysis of molecular weight?
Okay. Well, multiple scattering is usually not relevant to monoclonal and high concentration proteins. It turns out that because the cross-section for scattering actually decreases as you go to much higher concentrations. So multiple scattering is generally not an issue. There is thermodynamic non-ideality, and that's very important. And so if you take a single measurement, you will not get the correct molecular weight. What you need to do is measure as a series of concentrations and fit that to the relevant equations, which the CG-MALS system, Calypso system, does have in its software. And then you can determine both the molecular weight, any self-interaction, including repulsive and attractive interactions.
Great, thank you. Now, Dan, what if you have fluorescence in your sample? Does this affect the results? The listener is specifically referring to measuring particle size using DLS.
Oh, okay. So yes, it does. So if you have fluorescence, that will what will happen is you'll have a background signal. So remember that in DLS we're looking at the fluctuations in scattering signal, and so the fluorescence is usually not a fluctuating signal. It's usually fairly uniform in time, but it is a high background, and so it decreases your signal to noise, makes it more difficult to measure. What we have is we have a fluorescence blocking filter that can be inserted into the DLS collection fiber, and that eliminates most of the fluorescence and allows you to make good measurements.
Great, thank you. Now, is the Traos-LS detector suitable for protein conjugate analysis?
Absolutely. The Traos covers molecular weights up to about 10 megadalton, and used in conjunction with an HPLC UV and an Optolab refractive index detector, the three of them will address conjugate analysis, yes.
Great, thank you. And what is the level of resolution in terms of molar mass difference that the MALS detects?
So in terms of the MALS measurements, you can see differences on the order of a couple of percent. Now, if you want to actually resolve those species, if you have different species, the question is, can you separate them well enough? And usually, size exclusion column or even FFF will not give you a clear separation between species that have a couple of percent difference in molecular weight. But you will see, if you have a larger degree of polydispersity, you'll see that separation. You'll see the change in molecular weight across the peak. So you can see changes of a couple of percent, but you may not be able to resolve them in separate peaks.
Great, thank you. Can the MP-MALS be run on any type of sample matrix, for example, high salt or surfactants?
Generally, yes. So high salt, up to hundreds and hundreds of millimolars of salt. We've measured up to a molar of salt. Surfactants, yes, as well. In order to address the high salt, you need the extra accessory, which is the Atlas, which suppresses the electrolysis effects. But you actually can make very good measurements, yes.
Great, thank you. Could you discuss how precise the high-throughput DLS method is?
Well, it depends really what you want to measure. So let's say you want to measure the impact of formulation and you have some small aggregates forming. You can look at the polydispersity of the peak, and you can look at the average position, and you can clearly see shifts in the average position on the order of a couple of percent, say 1 or 2%. So that's the addition of just a few percent of small aggregates. Again, the polydispersity also is quite sensitive to that, and you can see a change by, excuse me, by a few percent. And these are robust, so you can make these measurements repeatedly. Because you have the option of putting in many replicates, you can see, you can also assess your precision and your error across the different replicates. And if you have, you can basically go up and down temperature, and you can see, do you recover the original situation? Now, if you're looking for a very precise measurement of size, say of a nanoparticle, the precision on that is something like 15 to 20%.
Great, thank you, Dan. As I look at the clock, I realize we're running short on time. There is quite a large number of questions that we haven't got to, but I will forward those to you, and you can respond to these folks directly. And that brings us to the end of today's webinar. If you have any further questions, please consider reaching out to Dan directly. His contact information you see is available on the screen. Just a reminder that today's webinar was recorded, and you will receive an email notifying you when the on-demand video is available. On behalf of Lab Manager, I'd like to thank Dr. Sohm for joining us today and Wyatt Technologies for sponsoring this Product Spotlight webinar. This concludes our Product Spotlight. I'd like to thank all of our attendees for taking time out of their busy schedules to join us today. We hope you can join us again for our next Product Spotlight, entitled Tips and Tools for Speeding Up the Pace of Chemical Development Innovation. This webinar will be occurring Tuesday, April 21st, from 1 to 2 p.m. Eastern Daylight Time. For more information, please visit our website at www.labmanager.com. Thank you, and enjoy your day.